Hi, everyone. My name is Rebecca Nugent. I'm the Senior Director of Biopharma Product Strategy at Twist Bioscience, and I'm so excited to talk to you today about tools for the good fight. Twist Bioscience is a leading manufacturer of synthetic DNA. We write DNA with the power of silicon. So on the slide that you see here, you have the Twist silicon platform compared to technologies where other competitors write DNA. And so what we do really well at Twist is that we've miniaturized DNA synthesis using a silicon platform. And so a footprint that's approximately this size can make 9,600 genes, whereas our competitors take that same footprint and can only make one gene. With the scale and throughput, we can drastically change the cost of DNA synthesis while continuing to have the same quality and decreasing the speed. And so what can Twist do for you? We can do precision DNA synthesis at scale. And with this core platform or core competency of DNA synthesis, we've created a wonderful portfolio of products, including products for synthetic biology applications like genes and oligo pools and libraries. And then we've gone into different markets, including products for next generation sequencing, including target enrichment, for DNA data storage, and then also for biopharma applications. And so what I'll be telling you today is a story of harnessing the power of the immune system and then highlighting how these tools, these different products, can help enable your research. So harnessing the power of the immune system. In the schematic here, we look at a patient who may have cancer. And to cure this cancer, to treat this cancer, T cells are extracted from the patient's blood, and then T cells are engineered to detect tumor, and then they're reprogrammed to be super killers of that cancer. Through a variety of techniques and a lot of biomanufacturing, researchers can grow an army of those cancer-killing T cells, put them back into the patient, and then basically the patient's own immune system can kill their cancer. And so when I look at this schematic, I see opportunities for engineering. And so today we'll talk about two different cell engineering constructs, chimeric antigen receptors and T cell receptors. And I'll go into what those are and how we think about it at Twist Bioscience. So engineering chimeric antigen receptors. Chimeric antigen receptors, or CARs, exist on the outside of T cells. And what they do is they basically find different antigens, targets, bind them, and then the T cell is activated to go and kill that specific cell. When I think of this, I love this figure because it shows a blueprint of what a CAR design is. And you see all those different domains. You see a binding domain, a hinge region, transmembrane domain, co-stimulation, activation, etc. As a molecular biologist, I think in DNA. And so let us translate your protein, your final construct, into DNA. So when I see this blueprint, I think of opportunities for engineering. I think of opportunities of isolating those different domains and then manipulating them. So here I see the binding domain, specifically the SDFB, or the variable heavy and light chain regions of an antibody, as an opportunity to do high diversity engineering, either through combinatorial variant libraries or through our biopharma discovery services. I look at this hinge region, which is shorter and flexible, and I immediately think of single-stranded DNA and oligo pools. So how can I create diverse sequences that can be engineered to create that exact environment to give your CAR success? And then transmembrane domains with those different genes, those different elements here, how can we mix and match them to create the system that works best for the system, for your cell? So Twist Bioscience's expertise in complex DNA assembly and libraries means that you can focus on the protein. And I'll highlight the combinatorial variant libraries and our biopharma discovery services. So what Twist Bioscience does really well is, again, using that high throughput DNA synthesis, we can precisely control library fabrication. And so while other approaches seen here may have weaknesses or biases when it comes to creating diversity, we can print every single diversity, every single sequence, and then put them together in a specific, precise, and bias-free way to introduce codon variants at precise locations. And we can do that through introducing it at a single site, multiple sites, stretches, etc. We also do MGS verification on those libraries, which means we know what's going into your system. So when you put that into your system and you see what comes out, your negative data is just as positive or just as useful as your positive data. These precision libraries give you control. You can do base by base precision, codon usage control. You can change the length of your CDR regions for your highly variable regions, for your heavier light chain antibodies, and you can basically make it so it's successful in your system. You can avoid restriction sites, you can do multiple germline scaffolds. Really, you choose what you'd like, and we can build it for you. Again, you're a protein, we'll do the RNA. So combinatorial variant libraries here, this is just a schematic of a specific region that has different mutations, and those mutations are highlighted in a different color. And you can see that the amino acids within that position change. Either they're every amino acid, or in this particular case, there's the majority of Gs. And so what we've done in this schematic is that we've highlighted a specific region, and we've highlighted what the amino acid changes are and the total diversity for this stretch. And using next generation sequencing, we can highlight how specific our system is. So this is the amino acid position on the top. This is the expected ratio of each amino acid within that position. And after NGS, next generation sequencing, this is what observed. And so you can see is that our system is very good at precisely putting in mutations, even if it's one mutation at a certain site, two mutations, or a combination. So let's say you would know your antibody and you know how you want to engineer your scFv, but you don't necessarily want to do the work. Well, Twist is committed to your success in antibody engineering. With our biopharma division, what we can do is antibody discovery and optimization for your target. We can do that with a breadth of complex libraries. We have a broad DNA buffet. And we have libraries of libraries that I'll go into in a second that basically creates a diverse portfolio of different types of libraries that you can go after to find what binds your specific target. The quality of Twist Bioscience is that we're experts in DNA synthesis, so the DNA libraries going into that discovery service is high quality DNA. And then we can do this with greater speed through miniaturization and automation, applying what we've learned in our gene synthesis pipeline to our discovery team. So we have deep technology for discovery and optimization. We have a broad buffet of libraries, including the hyperimmune library, VHH libraries, an AI hypermutated scFv, et cetera. And so let's say you either want to use our libraries of libraries, or you have an antibody that needs to be optimized. We also have our TAO, our Twist Antibody Optimization Platform, which takes your antibody of interest, we optimize it, we screen against your target, and then we deliver to you a fully human optimized lead. I'd like to briefly touch on engineering T-cell receptors. So if there's chimeric antigen receptors, there's also T-cell receptors. And here's a schematic of a T-cell receptor. Basically, there's a variable alpha and beta region that are connected by constant regions. And then signal goes into the outside of the cell for signaling. And so common elements for optimization and diversity for T-cell receptors are those variable alpha and beta genes. And there's also, if you understand your construct of interest, you can use gene synthesis or MaxiPreps to produce and synthesize your TCR construct of choice. But I'll briefly touch upon combinatorial assembly. And so combinatorial assembly is basically creating the diversity of genes. And so you have different parts, and then we can mix and match those different parts. And how they differ from combinatorial variant libraries is that we use gene inputs instead of smaller inputs. And so what we can do for combinatorial assembly, specifically for TCR discovery, is create a library of variable alpha and beta genes. I'm a scientist, so I always love to look at the data. And what I'm showing you here is how precise we are, how we can take our experience in combinatorial DNA assembly and how that combination assembly results in very little bias. And so if I look at the library going before cloning and then after cloning, we see pre-cloning reads here, post-cloning reads here. They're highly correlated, which means there's no diversity or no bias introduced when we clone. And then when we look at the uniform variant frequency, if we take this example of 4 input pools and a diversity of 150,000 different combinations, you can see that all of the dots are very closely together, which means it's very uniform. And that's important because a uniform library means that you have that sequence of each individual sequence is represented about the same amount of times. So when you put it through your screen, you know whatever comes out at high diversity or low diversity is because of the biology, because of your screen, and not due to the quality of your library. I'd like to highlight just one basic story, which is a tool for cancer genomics. And sometimes the right tool doesn't exist, and Twist can help with that. So in this schematic here, it's researchers from MIT developed T-Scan, which is a lentiviral delivery of antigen libraries into cells for endogenous processing and presentation on MHC molecules. The problem is that they needed a large number of 400 base pair genes. The tool didn't exist. Oligo pools, which have diverse amount of sequences, were too short, and gene synthesis of, I think, 250,000 different genes was just too expensive. So we worked with them to create a high throughput method for creating pools of non-clonal genes. We delivered them 250,000 different gene sequences within a single pool. And the result of that was this amazing publication, which I encourage you to read. So with that, I'm going to stop and ask the question, how can Twist enable your research? We're committed to providing tools for the good fight. We want to help enable your research so you can do the hard work of identifying tools, you can do the hard work for fighting cancer for your specific research. So I encourage you to bring us your need, contact us. We'd love to hear from you.